Okay, now we're going to talk about how bacteria change in their genetics. Gets to be real interesting. Okay, so we're going to look at mutations of genes. All right, first of all, a mutation is a change, as it says, in a nucleotide-based sequence of the genome. It is rare. It is not frequent. It is almost always a bad thing for that particular organism. And it can rarely, but it can lead to a protein that improves the organism's ability to survive. I mean, some mutations do help, most of them don't. In terms of frequency of mutations are pretty rare. Otherwise, the organisms, couldn't effectively reproduce if we were constantly mutating our genes. I think our species just wouldn't survive. Okay, so you can see the error rate. There are substances called mutagens that will increase the mutation rate. Most of these mutations stop transcription or code for most of the time a code for a non-functional protein, something that just does not work. And then that protein, when that as you make more copies of it, you get higher and higher levels of that protein, and it usually kills the organism. Okay? And you can see this positive selection for mutants. Just an example of this. I mean, you've got penicillin-resistant cells and penicillin-sensitive cells. And you would put this out on an agar plate, and this is a medium with penicillin, and it only grows penicillin-resistant cells. And then a medium without penicillin, so that should grow everything. And if you take these, and eventually what will happen is you take that mutagen, and you will begin to see that you will grow more and more and more of these organisms that are penicillin resistant. Once these start to divide, if the mutagen induces those mutations, it will start to divide, and you will get more and more and more of these organisms. And that's what happens. All right. Bacterial genomes can be changed through what we call recombination. It's basically where you take a segment of the DNA and it can be moved. It originated in one bacterial cell and it can go into a second bacterial cell. There are ways to do it. We're going to go through this. And after that, it's called a recombinant cell. It happens frequently in many varieties of bacteria. And it can involve any portion of that bacteria's genome. And remember, bacteria is only one circular piece of nucleic acid. So it's not like, it's, you try and recombinate a human, you got a problem because we've got a lot of genes. We've got a lot of chromosomes. We only have a single chromosome in bacteria, so that makes this much easier, okay? And this gene recombination is a major way that bacteria achieve genetic diversity and change, and is a real problem with resistance to our antibiotics and antimicrobials. Okay? It can be horizontal or vertical transfer. Vertical transfer means you're passing your genes to the next generation. You have a kid. Your genes are passed to your kid. That is vertical gene transfer, okay? Bacteria have the ability to do horizontal gene transfer, which is you have a donor cell, and the donor cell gives the genes to the recipient cell. And this is done in 3 ways in a bacterium: transformation, transduction, and bacterial conjugation. And you do need to know these, okay? So transformation, this is the first one. This definition is the recipient, the cell receiving the DNA, uptakes free DNA that's been released into the environment when a bacterial cell dies. So you get a bacterial cell that dies and lyses, and that DNA is out in the environment, and this recipient cell, close enough, can go and pick it up. Okay? The DNA exists as fragments, and the bacteria are able to take this up and they undergo transformation. And if the bacteria can do this, not all bacteria can do this, but some bacteria are very good at doing it. We call it a competent bacteria. Streptococcus does it, Haemophilus does it, Haemophilus influenza, Neisseria, i.e. Neisseria gonorrhea. Okay, so some of these bacteria are really good at picking up things out in the environment. This plays a significant role in the development of antibiotic resistance, okay? And basically able to get genes into these organisms that code for factors that allow that organism to cause disease. So some disease, we'll talk about this, some disease factors that can be inherited. An example of this is a famous experiment done by Griffith, and this is the transformation of Streptococcus pneumoniae. And you put basically a non-virulent strain, and you put it in a mouse, and the mouse lives. It's a non-virulent strain. You take the smooth strain, the virulent strain, and you put it in the mouse, and the mouse dies. But if you heat kill the smooth strain, i.e., we just talked about heat killing, all right, we heat kill the smooth strain, and we inject that smooth strain into the mouse, and the mouse will live because that smooth strain has died. Now, however, if you put the rough strain together with the heat-killed smooth strain, the mouse will die because Streptococcus is competent and it can pick up, again, the gene sequences from that rough strain, from these can be exchanged in terms of the smooth strain can interact with the rough strain and form, and basically, again, you've heat killed it and it shouldn't work. But when you put it with the other rough strains, which are perfectly good, then the mouse will die. Okay? All right. So let's look at these different ways of doing this. That's transformation. You just pick up. So in other words, this little strain picked up some sequence in here that the smooth strain could use, again, to kill the mouse. Okay, so that DNA was picked up in that environment and it made this virulent again. Transduction is when we transfer DNA from one cell to another using a virus. Okay, the virus has to be able to infect both the donor and the recipient. If you have a virus that infects a bacteria, it is called a bacteriophage or a phage. Sometimes you may hear people call it a phage. Same thing, okay? Wherever you put the syllable. There are two different phases, the lytic phase and the lysogenic phase of transduction, okay? The lytic phase, this is what we call generalized transduction. Lytic means you got to lyse, you got to burst the cell. So you have a bacteriophage that infects the bacterium. It hijacks that host organism's ability to replicate DNA, breaks down that chromosome into fragments, and during the assembly of that virus, it inserts, in the replication of the bacteria, it inserts its viral nucleic acid into that bacteria. So the phage is mistakenly packaged with the bacteria instead of the virus, instead of normal. So that viral DNA ends up in there, okay? When the bacterial cell lyses, this phage is released, this phage is released. When it infects a new bacterium, it injects the bacterial DNA into the host where it can be integrated. All right? Because the bacterial DNA is chosen at random, any gene can be transferred. I've got a little picture down here, so I'll show you in a second. The lysogenic phase, you basically use what's called a temperate bacteriophage that infects a host cell, integrates its DNA directly into the chromosome, becoming a prophage, and then environmental factors stress the virus to exit the chromosome and enter the lytic phase. Okay. The newly replicated viruses carry both the viral and the bacterial. So let's take a look at this. There's your lysogenic and your lytic, and I think this probably describes it better than just talking. So you have this you got the circular chromosome. There's your little virus. Okay. The virus comes in and is right there, is starting to inject its viral DNA in there. So it injects it into the chromosome. Okay. In the lytic phase, that phage DNA directs the synthesis, takes over the machinery of that bacterium and causes it to make new phages. And then as it fills up, the cell lyses and releases these phages. Okay? And the new phages can bind to another bacterial cell and start this all over again. So basically it gets infected, if you want to think about that, causes the cell to replicate that viral DNA, and you make more and more and more of these viruses. Okay? You can get, as we mentioned, this lysogenic if you have a temperate bacteriophage. Some of them are temperate, some of them are not. You can go into a lysogenic cycle. So once the phage DNA gets in, instead of the machinery being taken over and you producing just these viruses, the viral DNA gets incorporated into the bacterial DNA. And there you have a prophage. And then that prophage gets copied and copied. Okay. And exposure to stress, such as UV light or whatever, will trigger the removal of this piece from that chromosome. Okay. All right. So this is a real easy way when a virus comes in that it can force the organism to just make more and more and more and more and more viruses. Okay. It's really what it amounts to. All right. So you got to have a virus for your transduction. Okay. You got to have this virus. If you don't have a virus, they ain't doing transduction. Something that's commonly done in bacteria is conjugation. And we call this, basically, this is bacteria having sex. They're exchanging the genetic material from one bacteria to another. Okay. It requires physical contact between the donor and the recipient. The donor cell remains alive. And it's used by a pili, short little hairs. It's a conjugation or sex pili, and it transfers genetic material from one bacteria to another. You can transfer a lot of bacteria this way. A lot of genetic material. So you basically have here's an example. You can see I'm going here, but can you see the little pili? And these guys are connected and they are exchanging that material. Go into it in a second. Okay. What you exchange are called plasmids. And these are small, and you can exchange plasmids. You can explain, and basically a plasmid is, if you remember, are small pieces of extra chromosomal DNA within the cell. You have that single circular chromosome in the bacteria, but in the bacteria, you also have plasmids, okay? And they're little pieces of extra chromosomal right here DNA that is physically separated from our chromosome, okay? And it can replicate independently. This becomes very, very, very important when you get talking about plasmids that connote inhibition of your antibiotics. This is how you can exchange resistance plasmids, is what we call them. So these resistance plasmids are basically containing the information on how to allow that bacteria to be resistant to a particular antimicrobial. And bacteria can exchange that. So it is a real way of how a bacteria that is not resistant to an antibiotic can become resistant to an antibiotic. Okay, it can do it through exchanging resistant plasmids. Okay. All right, we have a couple types of plasmids. Fertility plasmids, and these are transferred via F cells in the conjugation pili, and resistance plasmids, and this was first found in antibiotic bacteria. Okay, so this is where we see it. And this is how it works. Now, this is the fertility, but it works the same thing with the resistance. So you have basically, here's your donor cell, there's your chromosomal DNA, there's your little plasmid. So this could either be a fertility plasmid or this could be a resistance plasmid. Doesn't make a difference. It's got some extra information. Produces the pili, extends it to the cell that is that does not have the plasmid, so it's considered to be F negative. All right, and you can see it again. There's that pili connecting the two bacteria. And it connects, and you replicate and transfer this plasmid through that pili. And then when they separate, the original donor cell is still F positive. Now you have a new donor cell that has changed from being F negative to F positive and can now share with another bacterium. That's the way it works. Okay? Well, so the difference is, I just summarized this for you to make it easier for you. Transformation picks up free DNA in the environment from a competent recipient. Transduction, you've got to have a virus. You've got to have a bacteriophage. And conjugation, cell-to-cell contact via a plasmid. Okay. And so I've also got a diagram that shows you the difference. All right. There's your little conjugation with your conjugation pili, and you share. And there's your transduction with your little viruses, and there's your transformation. Okay. All right. So that's it. That's basically how you can genetically change, how genetic changes occur in these bacteria.